Hello everyone, welcome back to my video of genetics. Today we are going to see about the experiment conducted by two scientists, Alfred Hershey and Martha Chase. The two scientists, they contributed to genetics to prove DNA is the genetic material undoubtedly. So when the experiment conducted by other scientists like Griffith, there was some controversy about the genetic material can be DNA or RNA or protein. So here they wanted to prove it using a kind of virus which infects bacterium, and what is the genetic material is going to be entered, what kind of genetic material is going to be entered to that bacterium to infect that bacterium, thereby to prove DNA is the genetic material, not the protein. So that is the conclusion of this experiment. After doing the experiment, they reached in the conclusion DNA is the genetic material. So when there is a question related to who are the scientists or who is the scientist undoubtedly proved DNA is the genetic material, you should give the answer as Hershey and Chase. Don't give any other name of the scientist. Okay, so let me explain this experiment. Before going to the experiment, you should know the structure of a bacteriophage. What is a bacteriophage? A virus which infects a bacterium. Virus infects a bacterium is called as bacteriophage. It is bacteriophage. Phage means virus. Okay, and bacteriophage means the virus which infects the bacterium. So he selected T2 bacteriophage for this experiment. So the name of the virus is T2. So the 2 can be written as subscript. T2 bacteriophage virus. The virus is infecting Escherichia coli bacterium. Most of the experiments in genetics is carried out in this bacterium. It's known as E. coli, or so we can write in short form E.C small letter coli. Escherichia coli or E. coli. So T2 bacteriophage is the name of the virus which infects the bacterium Escherichia coli or E. coli. Okay. So here we are going to check. So what was the experiment conducted by Hershey and Chase? They wanted to prove DNA is the genetic material by doing this experiment using T2 bacteriophage and Escherichia coli bacterium. So if you take the structure of a bacteriophage, here the structure of bacteriophage is given. So if you take the structure of bacteriophage, this is a hexagonal structure. Hexagonal means 6. Here you can see 1, 2, 3, 4, 5, 6. So 6 parts are combined together to form hexagonal structure, and there will be a collar. There will be a collar like structure, collar, and carried out with sheath. Okay, so if you take this structure, this is the head part, and this is the neck part, and this is the, and we are proceeding to the tail part. Okay, so these are the sheath. Okay, after that this will be going like this, and finally there will be a plate like structure. Okay, so here this is the tail plate, and the tail plate is making the attachment to the tail fibers. Okay, so this is the structure of bacteriophage, and here this head part contains the genetic material DNA in the case of most of the viruses, but RNA in the case of tobacco mosaic virus and retroviruses, like those viruses, RNA is the genetic material. Okay, so this is the structure of bacteriophage. So the virus which infects the bacterium. So here, once it is making the contact with the bacterium, this is the structure. This is making a contact with the bacterium. Here we are not showing the entire parts of the sheath. Okay, so this is making the attachment like this. Okay, so here there is genetic material. So when this is happening, the first thing is the tail fibers are making attachment to the bacterium. This stage is called as infection stage. So the first stage is infection. So infection means the virus is making the attachment to the host bacterium with the help of the tail fibers, and this is done in terms of virology. The term is called as adsorption. The term is adsorption. The absorption is a kind of infection is happened here. The virus is making the attachment to the membrane or the outer surface of the bacterium. Here it is E. coli. So after making the attachment, here it is digging the plasma membrane. Okay, in simple terms if you say, it is making a kind of penetration on the plasma membrane of this E. coli. So here, because of that, this is making a kind of a provision for the entry of the genetic material to the inside part of the host bacterium. After that, this is releasing the genetic material to the host bacterium. So the next stage, you can see with the host bacterium, the genetic material what is entered here from the virus is going to multiply and making many viral particles, and these particles are called as virions. What is the name? Virions. Okay, so here the replication of the virus happens. Genetic material is getting multiplied by using the host machineries, because without knowing the genetic material has entered, the bacterium is providing all the nutrition, all the enzymes for the replication to this virus. Virus is getting multiplied and virus is making many progenies, and these progenies are called as virions. Okay, and this stage is called as, here the cycle is called as lysogenic cycle, and the cycle is called as lysogenic cycle. Later, after the maximum multiplication has occurred, this will be rupturing the plasma membrane of E. coli and releasing all these virions outside. So that is the last process of this lysogenic cycle. Thereby it is able to release all the virions outside. Each virion will be taking part in the same kind of process and making infection to the many bacterium. Okay, so this is explained in the beginning of this experiment so that it will be easy to understand this Hershey-chase experiment. Okay, so now we'll see how this experiment was carried out by Hershey-chase. So there was doubt about whether the protein is going to be entered to the bacterium or DNA is going to be entered. So what they did, these both materials like protein and DNA, both are radio labeled. Okay, so when you take radio labeling, here if you take DNA, the composition of the DNA contains phosphorus 32. if you take the composition of protein, the protein contains sulfur. The protein contains sulfur 35. Okay. So these are the compositions of DNA and protein. DNA contains phosphorus 32. Protein contains sulfur 35. So here they allowed the infection of this T2 virus to happen to E. coli. That means they took E. coli and T2 bacteriophage, both were allowed to grow together in its medium, so that the virus was able to infect the E. coli and making the infection possible, and later we are going to detect whether the protein or DNA has entered to the virus. Okay. So for that, two types of medium they prepared. In the first medium, this is the first case and this is the second case. In the first case, they provided sulfur 35 radio labeled. Okay, they provided radio labeled sulfur 35 to the medium where T2 bacteriophage and E. coli are growing. Okay, so in this culture, T2 is present, E. coli is present, and in the nutrition we are providing the nutrition supplements what is given to this medium. Here we are providing sulfur 35 radio labeled. And second case, T2 is there, bacteriophage T2 is there, and E. coli is there, and here instead of sulfur 35 we are providing phosphorus 32 radio labeled. Okay, so radio labeled 32 phosphorus is provided in the second case, so that the sulfur 35 will be taken up in the formation of the protein capsule. So there is a protein capsule. Here capsule is a name we are using for bacterium. Here we are using the word capsid. What is the name? Capsid. Capsid means the protein sheath or the outer coating of this membrane, or the DNA is having a protective shield or protective covering in the virus, and it is called as protein sheath or capsid. So to make the capsid of this bacteriophage, the sulfur 35 was used by T2. Okay, and to make a DNA, the phosphorus 32 was used up in the second test tube. So here the protein sheath is radio labeled. Second case, the DNA is radio labeled, because here which is the composition of DNA, here it is phosphorus. Which is the composition of protein, that is sulfur. So the protein is radio labeled in the first case and the DNA is radio labeled in the second case. Okay, now after doing this, that is the stage is called as infection is carried out here. So the same thing is going to be displayed here. This is the first case. Okay, if this is the first case, we are going to see only the first case here. In the first case, we can see the absorption process or infection process, and here what is going to be injected also we are going to see. Okay, the first case, what is which material is a radio label? The first case, it is sulfur 35 is radio labeled. That is, if you take this is radio labeled. The capsid is radio-labeled with sulfur 35 and DNA is not radio-labeled. Okay, protein is radio-labeled. Here this is the capsid. Protein is radio-labeled because the composition of protein contains the sulfur 35. So this is getting infected, so that the second stage is the DNA is going to be injected and capsid, the protein sheet, will be staying outside the bacterial membrane or bacterial body. So in the next case, if you see, if this is the bacterium, we can see the DNA is entered. Okay, DNA is entered and the capsid is staying away from the bacterium with its tail fibers and tail plate. So here you can see the genetic material is not present in the structure of this virus, which is staying outside the bacterium, but the bacterial body or inside part of the bacterium, the cytoplasm, here we can see the genetic material. So this process is carried out here. Now blending. So this stage we are calling as blending. So here blending means we are trying to make the detachment of this capsid along with the other structures of virus after the entry of this genetic material, and we are going with centrifugation process. Centrifugation process, we are taking a vial, there we are adding all these contents, so everything is transferred to a vial. Vial means a small test tube which we are using in the centrifuge. Centrifuge is, we are using RPM, we are setting the RPM, rotation per minute, so that it is making the centrifugation, agitation, and according to the density, the contents what is added to the centrifugation equipment is getting separated in the vial. So the density, based on the density means the lesser weight things will be in the supernatant. Here this capsid is visible in the supernatant and this is radio labeled with sulfur 35. No? So we can see the supernatant is radio labeled and the genetic material, the DNA, which is seen at the bottom because of its heavy weight, and this is not radio labeled because in the first case we provided only radio labeled sulfur 35. It is not a part of DNA, it is a part of protein. So from this we are going to prove. So what is the conclusion of the experiment or interpretation of the result? The experiment was conducted by making the T2 bacteriophage infecting the E. coli, and after the adsorption or infection, we can see the genetic material is inserted and the other parts like the protein coating or the capsid along with the tail fibers and tail plate is staying away from the bacterium by blending process, and only the genetic material is entered and it is making the replication and many viral particles are formed, and that is happening here if you leave for more incubation period, and later we are doing the centrifugation process to isolate these two parts separately according to the density gradient centrifugation. So we are able to detect the radio labeled parts are present in the supernatant, not at the bottom of the vial. So that is proving the protein coating is not doesn't contain the genetic material. Protein is not the genetic material. Okay. So next we are going to see the second case. So what is the second case? Here the E. coli is there. E. coli is there. the same T2 bacteriophage is making the entry. Only the genetic material is going to make the entry. Here genetic material is there. Here this genetic material is radio labeled with phosphorus 32 and the capsid is not radio labeled because we allowed this bacteriophage to grow in a medium provided with phosphorus 32. This is second case. Phosphorus 32. So DNA, while making the DNA, the phosphorus 32 was taken up. So the radioactivity is seen in the genetic material, not in the capsid as in the first case. First case, capsid, radio-labeled capsid. Here it is radio-labeled genetic material, and same process is going to be continued. Adsorption or infection, then blending. The same blending will be happening here. So if this is the case, this is the bacterium. The genetic material is entered. So the genetic material is radio-labeled. Okay, and here outside, what is staying? The same like the capsid protein, the protein sheath or the capsid with along with the tail plate and tail fibers, and the blending will be carried out. Blending will be carried out, and the last process is called as centrifugation will be carried out to separate the bacteriophage other parts which has not entered to the bacterium, so that in the vial, centrifugation vial, after centrifugation, by setting the RPM, we are checking the supernatant or at the bottom which is having the radioactive material. So here we are seeing non-radioactive material. Here radioactive material. What is the interpretation of this experiment? Because here the DNA is radioactive because of the presence of phosphorus 32, because sugar phosphate backbone is there in the skeleton of DNA. So here we can see DNA is, the bottom part is radioactive and the top part, that is supernatant, is there is no radioactivity is visible. So from this it is concluded as the DNA is the genetic material that has entered. So what is the reason this is heavy? The reason it is heavy here, it is because many, the content wise, if you see, this is only the one viral particle except the genetic material. Here the whole content of the E. coli is present along with the virions. So this will be heavy. That is the reason we are seeing this is sedimented at the bottom of this vial because of the heaviness, and radioactivity is visible there because of the presence of phosphorus 32. Okay. So from this the conclusion was DNA is the genetic material, not the protein. Undoubtedly it was proved by Alfred, Hershey and Hershey-chase. Thank you.